Ribosome, the protein assembly factory of life. The Nobel Prize in Science is one of the most prestigious awards, honoring inventions and discoveries that have changed the world. In the fields of biology and chemistry, there is a special achievement that has been awarded the Nobel Prize, the discovery of the ribosome, the tiny machine of life. But what exactly is a ribosome? Why is it so important that it won a Nobel Prize? And what is the secret behind the ribosome's operation? Let's explore more deeply the miraculous mechanism of the ribosome, the protein-producing machine in every living cell. Imagine your cell as a miniature city. The cell nucleus containing DNA is the library that stores the entire blueprint of the city. When a certain structure needs to be built, that is, a specific type of protein, a copier, enzyme, creates a copy of that blueprint called mRNA, messenger RNA. This copy is then sent to the factories in the city to carry out the construction. The ribosome is that factory. The core function of the ribosome is translation, reading the information on the mRNA blueprint and assembling amino acids in the correct sequence to form a complete protein chain. Proteins are the workers and building materials of the cell, performing almost every function, from creating structures, catalyzing chemical reactions, enzymes, to transporting oxygen, hemoglobin. Without ribosomes, the information in DNA would forever remain a useless blueprint, and life could not exist. 2009. Nobel Prize in Chemistry, illuminating the black box of the cell. Before 2009, scientists knew what ribosomes did, but did not know exactly how they did it at the atomic level. Ribosomes were like a complex black box. The work of Venkatraman Ramakrishnan, Thomas A. Stites, and Ada E. Yonath changed that. Using X-ray crystallography techniques, they succeeded in creating a detailed 3D map down to the atomic level of the ribosome. This was a feat because the ribosome is a huge and extremely complex machine, consisting of hundreds of thousands of atoms. The main discoveries from this 3D map include: A. The two subunit structure. They have shown exactly how the small subunit and the large subunit fit together. The small subunit plays the role of reading the genetic code on the mRNA, while the large subunit performs the joining of amino acids together. B. The mechanism operates at 3 key sites. The most important discovery is the identification of 3 main active sites on the ribosome, where the translation process takes place. A site, aminoacyl. The entry gate, where a tRNA transfer RNA molecule carrying a new amino acid enters and pairs with the genetic code on the mRNA. P site, peptidyl. The assembly workshop, where the new amino acid is attached to the growing protein chain. Position E, exit. The exit where the tRNA molecule, after having delivered the amino acid, leaves the ribosome. This mapping is like having a detailed blueprint of a car engine. For the first time, we can see each molecular piston and gear in action, how the mRNA slides through, how the tRNA brings the materials, and how the protein chain is produced smoothly. 1. The ribosome is not a protein, but a ribozyme. This is one of the most astonishing discoveries about the ribosome. Although it is made up of both protein, R protein, and RNA, rRNA, the catalytic heart of the ribosome, where the peptide bond is actually formed, is entirely RNA. This means that the ribosome is essentially a ribozyme, an RNA molecule capable of catalyzing chemical reactions, rather than a typical protein enzyme. The proteins in the ribosome mainly serve as a scaffold, helping to stabilize the structure and enhance the efficiency of the rRNA. Importance: this discovery strongly reinforces the RNA world hypothesis. This hypothesis suggests that RNA, rather than DNA or protein, was the core molecular form of early life. The fact that the ribosome, the most ancient and universal machine, relies on RNA to perform its most important function is compelling evidence that RNA once played a dual role, both storing information, like DNA, and catalyzing reactions, like protein. 2. Sophisticated quality control system. An accuracy of 1 error per 10,000 amino acids is not accidental. Ribosomes have extremely sophisticated quality control mechanisms to ensure that translation is as accurate as possible. Codon-anticodon pairing. When a new tRNA carrying an amino acid arrives at the A site, the small subunit of the ribosome checks whether the tRNA's anticodon correctly matches the codon on the mRNA. Kinetic proofreading. This is a two-step mechanism. Even when there is an initial codon match, the ribosome still has a short delay before forming the peptide bond. During this time, if the binding between the codon and anticodon is not strong enough, i.e., incorrect, the tRNA molecule is more likely to be kicked out than a correct tRNA. This is similar to trying a key in a lock. A wrong key may be inserted, but it will wobble and can be easily pulled out before it can turn. Induced fit. The correct recognition of the codon-anticodon pair causes a structural change in the ribosome, locking the tRNA into the correct position and activating the peptide bond formation process. Incorrect pairs will not be able to effectively cause this structural change. 3. Protein synthesis control center. Ribosomes do not operate mechanically. Their speed and efficiency are tightly regulated to meet the cell's needs. Polysomes or polyribosomes. To maximize efficiency, multiple ribosomes can simultaneously slide along a single mRNA molecule, like multiple train cars running on the same track. Each ribosome produces a protein chain. This structure is called a polysome, allowing the cell to quickly produce a large amount of necessary protein. Regulation according to environmental conditions. When a cell encounters stress, such as nutrient deficiency or high temperature, the overall protein synthesis process is usually reduced to save energy. The cell does this by phosphorylating, adding a phosphate group, to translation initiation factors, preventing ribosomes from assembling on mRNA. Conversely, when conditions are favorable, this process is enhanced. 4. The existence of specialized ribosomes. An emerging and intriguing concept is that not all ribosomes in an organism are identical. There is evidence for the existence of specialized ribosomes. Hypothesis: ribosomes in different types of cells, e.g., brain cells versus liver cells, or under different conditions may have slight variations in their protein or rRNA composition. Potential function: these changes may cause some ribosomes to preferentially translate a specific group of mRNAs. For example, a ribosome specialized in muscle cells may be more efficient at synthesizing structural muscle proteins. This creates an entirely new layer of gene regulation, allowing the cell to fine-tune protein production with extreme flexibility. 5. Role in common diseases. Besides rare genetic diseases, ribosomopathies, ribosome dysfunction is increasingly shown to be associated with more common diseases. Cancer. Cancer cells grow and divide uncontrollably, requiring a large amount of protein. Therefore, they often enhance ribosome activity. Some cancer treatments are being researched that target the inhibition of ribosome biogenesis to starve tumor cells. Neurodegenerative diseases. Alzheimer's, Parkinson's. There is evidence that errors in the ribosome's translation process can lead to the production of faulty, misfolded proteins. The accumulation of these proteins is a hallmark of many neurodegenerative diseases. In summary, the ribosome is not only a factory, but also an intelligent molecular processor. It is both a legacy of the ancient RNA world and a center for quality control and sophisticated regulation, playing a key role not only in basic biology, but also in human diseases. 6. Evaluation of effectiveness and benefits. Asking about the percentage effectiveness for people practicing this method may be a misunderstanding. The ribosome is not a method or therapy that humans can practice. It is a fundamental component of biology. However, if the question is interpreted as, what is the biological efficiency of the ribosome? And to what extent do the benefits of understanding it bring effectiveness? We can answer as follows. A. Biological efficiency of ribosome. The efficiency of ribosomes in nature is almost absolute. It can be said that the efficiency for life is 100%, because if ribosomes stop functioning, the cell will die immediately due to the inability to produce proteins. Speed. A ribosome can link about 20 amino acids per second in bacteria. Accuracy. The error rate is extremely low, only about 1 error per 10,000 to 100,000 amino acids. This is an astonishing level of accuracy, ensuring that the proteins produced have the correct function. B. Benefits from the Nobel Prize-winning work. Understanding the structure of the ribosome has opened a new era in medicine, especially in the field of antibiotics. Development of new generation antibiotics. Estimated effectiveness very high, with the potential to save millions of lives. Many common antibiotics, such as tetracycline, erythromycin, work by attacking and disabling bacterial ribosomes. The slight structural differences between bacterial and human ribosomes allow the drugs to target only bacteria without harming us. Previously, the discovery of new antibiotics was mainly based on trial and error. Now, with the 3D map of bacterial ribosomes, scientists can design drugs in a targeted manner. Rational drug design. They can create drug molecules that fit precisely into important sites of the bacterial ribosome, like a key fitting into a lock, helping to increase effectiveness and reduce side effects. This is an extremely important weapon in the fight against antibiotic-resistant bacteria. 6. Risks and disadvantages of ribosome malfunction. The perfection of the ribosome is very important. Any deviation in its function can lead to serious consequences. A. Risks from intrinsic errors, gene mutations, ribosomopathies. This is a group of rare genetic diseases caused by mutations in genes encoding ribosomal proteins or RNA. When the ribosome is defective, protein production is affected, leading to serious problems. For example, diamond black fan anemia, a rare blood disorder caused by bone marrow failure, is directly related to ribosome defects. Prevention. Genetic counseling for families with a history of the disease. Prenatal diagnosis. Currently, treatment for these diseases remains very difficult. B. Risks from external agents. Toxins. Some of the most dangerous toxins in nature act by directly attacking the ribosome. Ricin. An extremely potent toxin found in castor beans. Even a very small amount can be lethal. Ricin works by cutting an important bond in the large subunit of the ribosome, causing it to cease functioning permanently and halting protein production. Diphtheria toxin. Produced by the diphtheria bacteria, it also inactivates a factor necessary for the ribosome to move along the mRNA. Prevention. Avoid exposure to toxins. Get full vaccination to prevent diseases such as diphtheria. Develop antidotes based on understanding the mechanism of toxin attack on ribosomes. In summary, the work on ribosomes is not only an outstanding scientific achievement, but also a foundation for invaluable medical applications. It shows us the complex and efficient beauty of life at the molecular level, while providing powerful tools to fight disease. Support by liking the video and subscribing for the upcoming series. Thank you.